So hi, everybody. Thanks for coming. This is Science by the Slice. I'm Camilla Saunders. I'm the Science and Magazine. I also work with the RTP chapter of Sigma Xi, which is one of the sponsors of this event, along with Communicators of North Carolina. And we thank the NC Biotechnology Center for sponsoring our pizza, as always. So this series generally runs monthly during the academic year. So our next one will not be until January. So keep an eye out for the invites to that event. If you have not signed up, there's a way to do that through the Biotechnology Center website, or just let me know and I'm happy to have you. So today we're really excited to have a talk from Dr. Adam Hartstone-Rose, who is a professor of biological sciences at North Carolina State University and is specifically a comparative anatomist and functional morphologist. He is going to talk to us today about Winnie the Pooh and other stories about masticatory adaptation. Please take it away.
This is a camera on me?
I think, yes.
Okay.
Yes, but it doesn't.
It tends to wander a lot. So if I do something or people can't hear me or something like that, let me know. But feel free to stop me at any time to ask questions. Hopefully this can be fun and interesting. I give a lot of different types of talks and on a lot of different subjects. And this is one of they're modified from one of my favorite talks to give to audiences of multiple ages. So it's a good hook for kids and colleagues alike. But yes, I am Adam Hartstone-Rose, and I run what we call the Comparative Anatomy and Functional Morphology Research Lab at North Carolina State University. And basically this means that my students and I study how animals are adapted to do what they do. And we do this, generally we talk about this in terms of many levels. And specifically, we describe this as we study all of the levels of deadness. This is very appropriate for so close to Halloween. Like kids usually think I'm trying to talk about zombies or something like that. But actually, when we think about the levels of deadness, we categorize all of our research across a spectrum from live animal behavior, which is the least dead you could be, a lot of dissection work. That's probably what our lab is most famous for, looking at muscle anatomy, and I'm going to talk about that. So that's pretty freshly dead. And then more dead than that are bones and teeth. So in my lab, we have about 1,000 skulls and skeletons of different animals. A lot of them are the ones that we've dissected, but we also go to museums to collect data, et cetera. And then a great amount of the work that we do is also paleontological. So long dead, that's the most dead you could be, is so dead you're turned to a rock. And so we study things across this entire spectrum. Mostly we focus in my lab on mammals in general, but specifically we focus on carnivores and primates. Those are the two main groups that we study. And we study various aspects of their anatomy. Predominantly, we have some research looking at the limb anatomy to deduce like how, for instance, muscles are adapted for terrestrial locomotion or arboreal locomotion or things like that. But probably the majority of our research is on feeding adaptations, and that's really what we'll be talking mostly about today. So for instance, we'll do studies where we'll look at the analysis, or we'll do analyses of the diet of living species. So does anybody know what this animal is? Yeah, that's a snow leopard, right? And so we might figure out that snow leopards eat a certain type of prey animal. And then we will dissect members of this species to learn how the muscles allow for the consumption of that specific diet. And then we'll look at the attachment areas on skulls and the anatomy of teeth to learn about how the skull is adapted to allow the consumption of that kind of diet. And from that, we can then make inferences based on our understanding, our research on the skulls of extinct species, and sort of reconstruct what the soft tissues would allow, and ultimately get at things about the ecology of those species. And so this is a this meeting is being live streamed. Yes, okay. I will say okay to that. So this is a recent reconstruction that I did of Smilodon, the great American saber tooth. And ultimately what a lot of our research is interested in is trying to understand the ecological adaptations of extinct species. In this case, carnivore. We also really love lemurs. We are in the lemur capital of the world here in the Triangle. And actually, I came to this area, gosh, 4 years ago, because I was a lemur lover as a teenager. And so, of course, I went to Duke to study lemurs. And so we'll study extinct lemurs as well. All right, so this is kind of our little shorthand where we have the snow leopard. Oh, the laser just simply does not work on screen. We have the snow leopard up at the top, and then going all the way across to the saber tooth on the other side. And so I'm going to talk about a few examples of this kind of research. To be 100% honest, I'm not sure how long this talk is at this point. So I'm gonna get through it, and I'm really hoping that we can leave it. I have plenty of time in the end to have conversation. But yeah, like I said, just stop me mid-sentence if I've gone over it, or stop me if you have a question. All right, the behavioral work on live animals. The reason we study animal behavior, truly, like, honestly, my people who know me well, like, this is kind of a secret. We'll keep it between us and maybe all of the people online. But prefer dead animals to live animals. It's like a kind of a dirty secret. But the problem is that before we can understand how animals are adapted in terms of their anatomy to eat what they eat, we have to figure out what they eat, right? So by the way, this is also a hilarious talk to give at lunchtime, because I find that my audience starts getting like really paranoid when they realize like I actually study like how you are processing that food, and especially hilarious when people are eating fruit salad, which will become apparent in a minute why fruit salad specifically. Okay, so we have to figure out what they eat. In other words, I study functional morphology, but we need to quantitatively define that function. Now, there are many qualitative descriptions of what animals eat. So we know, for instance, that pandas eat bamboo, and there's like, you know, these eats fruits, or this species eats meat, right? But in order to say how they eat that food, we need to better understand the quantitative parameters. So how big is the food, right? That relates to how wide you have to be able to open your mouth and produce force at that gate, right? How obdurate, how mechanically challenging is that food to process, right? If you're eating, if you have to eat a Brazil nut and you don't have a nutcracker, how can you get into that nut, right? And there are some monkeys, for instance, that can crack like a really hard nut. That is an amazing masticatory requirement. What things like pandas do to eat bamboo, that's remarkable. That's something that we cannot do, and it's because our anatomy limits that. And so one of the ways that we tried to get at just quantifying one of these parameters is we came up with a concept that we call maximum ingested food size, which we abbreviate VB, the variable of this bite size. And this concept, so we're really wanting to know how wide an animal needs to open its mouth in order to ingest food. And so what we did was we came up with a very simple experiment. So maximum ingested food size we define as the largest block of food an animal will ingest without biting it apart first. So here's where we start to get at the fruit salad thing. Like if you were to get a really big piece of melon, you wouldn't probably think about it, but you might reach for like a knife and a fork to cut it apart, or you might take a bite of it. But if it was a smaller piece, then you might be liable to eat it whole, right? And that's exactly what we were trying to get at. And we started this, we designed this line of research first at the Lemur Center, and then we expanded it to many other places. And the experimental design is really simple. We would take different types of foods. So for instance, we might choose like cantaloupe during a day of experimentation. And we would cut that food to a very specific size. And we'd cut it into a cube, because a cube is a shape that you can be really consistent and repeated. And so we might cut on this day for this given lemur, we might feed this lemur 5 cubes of cantaloupe cut to, for instance, we can say that this is like 1 centimeter on each side of the cube. This is where I'll stop and I'll ask my students to tell me how, the volume of that cube. It starts out very easy, right? So 1 cubic centimeter. And then they're shocked to know that that's like 1 milliliter. And then we have to go back all the way to like high school. Yeah, so we'll feed it we'll feed it 5 cubes, for instance, 1 centimeter on each side, and we'll watch what the animal does. And we're looking for 2 things. So if this food is comfortable, is a small enough size for them, then the animal will pop each one of these into their mouth whole, chew it up, swallow it, then pop the next one. And then what do you think we would do the next day when we came back? So that was below the size that we were looking for. So we'll make it bigger, right? So the next day we'll come back and we might give it cubes that are 2 centimeters on each side. And again, now my students are starting to sweat because they have to figure out the volume of 2 by 2 by 2. Not that hard, guys. So, but this is a much larger cube, right? And at this point, we'll give it that same the same animal, the same type of food, but at this larger size. And if this size is beyond that VB, beyond that maximum ingested bite size, then what they're going to do is they're going to bite it apart first. Sometimes they'll bite it in half and then do both halves separately. Sometimes if we've guessed terribly and we've gone like way beyond it, sometimes they'll sit there and they'll eat like 8 pieces and we have to watch them like nibble on the little thing. And so the next time we'll give it a size in between. This is like that old guessing game, right? Where like I'm thinking of a number from 1 to 100. And there's a objectively smart solution to that. You just keep cutting it in half until you get it, right? And so the next day we'll go back and we'll give it cubes that are 1.5 centimeters on each side. And at this point, my students are really sweating when I try to them to tell me the volume. And we're looking for a point at which the lemur, so what do you think happens when we get to the size that they are more or less comfortable with? What do you think we're looking for? Like what would you expect? Yeah.
Half the people popping it in their mouth like that and half kind of taking a bite out of it.
Yeah, except so it differs by individual. So some like lemurs, like you'll have like one lemur that is just going to be like pounding food and another is going to be a little more dainty. So we're looking for that same ratio of 50-50, but within the animal itself for a given food. So what we're looking for is for this lemur to bite apart a few of these, but then to pop a couple of them whole, just like that, right? And then the next day we'll go back and we'll feed it that same size. And hopefully the lemur does the exact same thing and does 50-50. Unfortunately, live animals are so annoying. And so sometimes they'll like it's like based on, as my son would say, vibes, like how they want to process these foods. But ideally they're gonna bite apart half of them and eat whole the other half. And hopefully we get to the point where we have like 50-50 of our sample at a given size. It has processed this. And that's what we'll call the VB for that specific type of food for that specific animal. And we'll look at this for a few different foods. So we might give it sweet potato and carrot. Not all of our foods are orange, but it happens to be in this case very convenient. And we'll feed these to animals across dietary spectra. So some lemurs are more adapted to eating leaves. Some are more adapted to eating fruit, and then they exist across great amounts of body size variation. So little mouse lemurs all the way up to bigger ruffed lemurs. And so this is what it looks like when we do this. Hopefully all of y'all have spent time at the Lemur Center because it's a magical place. Lemurs are really great animals. They always look kind of freaked out, but they're really fun to work with. And so this is, does anybody know what kind of lemur this is? A red ruffed lemur, yeah, red ruffed lemur. And this is so that they're highly frugivorous. They love the juice from fruit. So they'll throw their head back and they'll try to swallow that antelope as much as possible. And this is my favorite species to talk about for this because an amazing thing happened. So these are all the different species that were at the Lemur Center at the time that we did this with. And we found a really cool thing. We found that, of course, larger lemurs eat larger pieces of food. That makes sense. But what's neat is that the lemurs that are adapted for eating fruit eat significantly much larger pieces of foods, of all foods, than the lemurs that are adapted for eating leaves. So we were talking about your folivores. Folivores are not going to be able to open their mouths as wide. And so in this case, these are 2 lemurs that are almost the exact same body size on average. So this is a cockerel shafak, and that's our red ruffed lemur. And this is the relative size of the cubes of food that they eat. And the reason that the red ruffs are near and dear to my heart is the remarkable fact that a red ruffed lemur can eat a cube of cantaloupe that is larger in volume than its cranial capacity. So without biting it apart first. So they will shove a whole piece of melon in their mouth that's larger than their brain, which is adorable. And it's important to know that that's both because they have an amazing ability to open their mouths wide, like a huge gape, and also lemurs are not that bright, right? So, you know, they don't have the greatest cranial capacity, but we love them anyway. Maybe we love them specifically because of that. So we then repeated this. We did this with all sorts of monkeys, and then we did it with gorillas. That was really fun to try to get huge cubes of foods to really push like what a 400-pound silverback gorilla was capable of processing. And we were able to document this across many different primates. And what's great is that we can then take this behavioral data that we have. So now we know the size of food that an animal is comfortable eating, and we can combine this with our dissection-based data. And we can see how the animals are adapted for this by analyzing their muscle anatomy. And I don't really have time to go into the details of this, but the thing that my lab really specializes in is what we call analyses of muscle fiber architecture. And in short, basically, you can take any muscle, and a muscle's abilities are based on two overarching variables, one being the fascicle length, so the length of the muscle fibers, and fascicles are just bundles of muscle fibers within that muscle, and also their arrangement, which tells you something about the other key variable, which is what's called the physiological cross-sectional area. And so what's neat is that fascicle length relates to the ability of a muscle to be fast or stretchy, and physiological cross-sectional area relates directly to how much force that muscle can produce. Okay. And so we can study how these muscles are basically put together, what the arrangement of these fascicles are. So some muscles have their fascicles running all the way from sort of the top to the bottom, and so that's the longest that arrangement of fascicles can be. And so long fibers equate to greater stretch. Okay. Other muscles are what we call pennate. So their fascicles are arranged, and it's called pennate because it looks like a pen, like a quill, right? Like feathers, right? And so, and when in muscles that are arranged like this, more fibers equate to greater stretch. Greater strength. So the longer a fiber is, the stretchier it is, the more fascicles you have, the stronger the muscle is. And so for a given muscle volume, pennation increases the cross-sectional area, but it decreases the average fascicle length. And so this is this fundamental trade-off that we see, that for a given muscle volume, muscles can be adapted for either maximal stretch or maximal strength, or some compromise in between. If you need to produce great force and also great gape, then you have no choice but to have larger muscles, right? Because then you have more fascicles that are also long. So things like hyenas that have to open their mouths for like a large prey item, but also like be able to crack bones, they just have to have larger muscles overall. All right. There are two ways that we've studied this. I always tell my students that I use like 18th, 19th century methods to do this, and I want them to use like the 21st century methods. So when I started doing this, we used to do what we call chemical dissection. So we would do gross dissection of the animals, and then we would disarticulate the muscles using acid. Another reason this is fun to talk about at lunchtime is because it kind of looks like tuna fish, but this is in like sulfuric acid, so it doesn't smell like something you want to eat. Although we've done this with vinegar, and that does smell delicious, but you still shouldn't eat lemurs. Okay, and so you can actually, when you break down the muscles, you can see the fascicle lengths. When I started doing this, I wasn't like an aficionado of North Carolina pulled pork barbecue, but that's what we're getting at, right? So when you do pulled pork, you're pulling apart the fascicles. So when you cook something low and slow, then it disarticulates along these boundaries like this. The much cooler way that my students have been doing this, that's much fancier, is what we call DICE-CT. And that stands for diffusible iodine-based contrast enhanced computed tomography. And all this fanciness means is that we stain specimens using some kind of reagent that changes the density of the tissues that we're attaching it to. In this case, it's iodine. So if you were to take like a CT scan of your hand or something like that, all that you end up seeing is you see the bone, you see the air, and then all the soft tissue is one uniform shade of gray, right? And so if you want to study like soft tissue injuries or something like that, like if you've done something to your knee or something, then you need an MRI because that will give you more definition of those soft tissues. However, this iodine will bind to the glycogen in muscle fibers. And when we do that, we can actually see individual fascicles in space. And so that allows us to do what we call digital dissection of these animals. So this specimen here was stained using this iodine. And then when we scan it, we can actually see these individual fascicles and isolate them. And now we can measure those fascicles in 3D space. And we can do fancier things like figuring out their angles relative to each other and whatnot, not just the lengths. All right, what this does, so what we found was that muscle architecture scales with food size in primates. So animals that eat larger foods, they have longer fascicles. So we were able to basically document that. This also means that we're able to then take like a specimen of some rare animal from a museum that we've never done a behavioral analysis to, and we can study its anatomy and make deductions about what it was capable of doing. We did the same analyses in cats, and cats are really fascinating. So we get back to our snow leopard. Cats are super cool because cats all eat vertebrate flesh like that. So they don't have the same kind of dietary diversity that lemurs do, where there's fruit-eating ones and leaf-eating ones. These guys all eat basically meat. We call them hypercarnivores. But what's awesome is that they vary in terms of the size of their prey, right? So really specifically the relative size. So some cats are adapted for eating animals that are larger than their own bodies. Like they'll regularly take down a big prey item. Some cats, cat species really only target like little animals, like little rodents, right? And they'll never approach like a big animal. So for instance, we have this pair of animals right here. Does anybody know what these guys are?
Check.
So, nope, not ocelot. I think I heard jaguar. This is a serval. Do you guys know what a serval is? They're really cool. It's not fair. I'm not showing you the ears are very notable, but the most notable part of a serval is that it's got very long legs. So maybe I threw you off. And does anybody know this one? This is a caracal. So it's kind of like an African lynx. And what's neat is that these are two species that are most closely related to each other. They're sister taxa. But the caracal is adapted to eating large animals, and the serval only eats small animals. They both eat kind of birds, but when really pushed, and they live in the same environments, and when resources are scarce, servals specialize in eating little itty bitty things, and caracals are known to take down like antelopes bigger than themselves, which is really amazing. And what we were able to show is that the fascicle lengths of their masticatory muscles reflects that. So caracals that eat much bigger animals than themselves have much longer fascicles than the servals do. And then the cats that eat things that are kind of intermediate in size and relative size fall in between. There's this one point very high at the top. This one here is this taxon. Does anybody know what this is? Has a great scientific name, Neophilus nebulosa. Nebulosa. So when something's nebulous, it's like, what is that? Cloudy? Yeah, so this is the clouded leopard, right? So this is a really interesting taxon. It's a kind of a medium-sized cat from Southeast Asia. And they are famous for having the longest canines relative to their body size. And so they also have to have really long fascicles because in order to open their mouths to clear the same amount of space in between these really long canines, they have to be able to stretch their jaws really far open. And so we're able to show that as well. And then we've shown that bite size and anatomy relate to food properties. So as you would expect, animals take smaller bites of more obdurate food. So like if we feed our lemurs the carrots and the melon on the same day, then of course they're going to eat much smaller pieces of carrot than they do the melon. And then their muscles are capable of producing more force. Right? So the animals that are adapted for eating the tough stuff, we find they can do that, of course. And then we take this even further where we then can project this from our muscles, interpret the bones, and then try to make deductions about fossils. So for instance, we take this understanding of the architecture of the living cats, the extant felids, and study how their attachments, how the muscles attach to bones, and how that can be interpreted in telling us in terms of stretch and strength. And so if you see all of these little dotted lines, these dashed lines, these are different proxies that we can use to try to assess how that muscle is spanning from the cranium to the mandible and that kind of space. And then we correlate those lines with the muscle architecture stuff, and we can then make predictions about the stretch and strength of these muscles in extinct cats. So for instance, we can then take this Smilodon, that American saber-tooth, and try to deduce how much could it open its mouth, right? Now, of course, this thing has to open its mouth really wide. If it only opens its mouth halfway, it's not getting any food into its mouth, right? And so we really don't understand how these jaws work. There is no living saber-tooth. And so trying to understand its abilities based solely on the bones themselves has stymied scientists for ages, because we don't have muscles of these extinct things. And so our research, we were able to reconstruct what the muscles were capable of in order to try to figure out how much it could open those jaws. And we found something kind of interesting. First of all, we found that Smilodon had a gape similar to modern cats of a similar size. So Smilodon was about the size of a tiger. And although it's angular gape, like it could open its jaws, actually some people have reconstructed this to more than 110 degrees, right? So more than a right angle opening. Like imagine if you could open your jaw, be like in your neck, that'd be crazy, right? So opening really, really wide, right? But the clearance between the canine tips is really almost the exact same as what you would find in a tiger, in a living tiger. So for a similar sized animal, they had a similar gape. The problem is that in order to accommodate this amazing ability to open the jaws really, really far in terms of the angle, it sacrificed bite force. And so it had a much weaker bite force than a living tiger. And in fact, its bite force was similar to that of a jaguar, which is an animal that's about half the size of a Smilodon. And so it kind of, so in one respect, we answered one of the mysteries about what it was capable of doing with its jaws, but then it opens up this whole other mystery that why would a huge animal like this, essentially this whole lineage evolved to sacrifice bite force to accommodate this amazing tooth, and we still don't really understand why it went down this road. And ultimately, maybe we could say it was doomed to failure because all saber-tooths are now extinct. What's amazing is this saber-tooth anatomy evolved independently at least 6 different times in evolution in mammals, and even in pre-mammals. So in what we used to call mammal-like reptiles, some of them had saber-ish teeth. There were marsupials that were saber-tooth, and then different types of carnivores, and of course the cats. And all of them are extinct. Yeah.
Is the leading hypothesis that it's like a mating display or something like that, or
That's a great question. So a lot of animals, especially like primates, have giant canines because of this sort of sexual display stuff. So like if you think about a baboon has huge canines, actually longer than a lion's canines. The males do, the females don't, but that's because of the social structure. One thing that's quite mysterious about saber-tooths is that they're potentially not sexually dimorphic at all. Like for a tiger or a jaguar, we can tell the difference between a male and female based on the skull alone. We can't determine sex on those. And so most animals that have the kind of mating system that would lead to that also have great divergence of sexual dimorphism. So we don't really know. A lot of people think that this canine allows these animals to kill large prey kind of independently, right? Whereas other animals that kill relatively large prey have to do it in a group. But the truth is, we really don't know. Another like strange mystery is that this long and scary looking tooth actually is like kind of fragile, right? So if like you went up to this animal and you punched it like from the side, right? They're extinct, so you don't have to worry about doing something dumb like saber-tooth. But if you pushed on this tooth, it's just a very long bar, right? So you know that if you have like a very long lever and you push it, it's going to be much more vulnerable than if you have a tooth like the length of a tiger tooth. And so that doesn't seem very conducive to struggling with large prey either. So, you know, when we answer one set of questions, it always opens up like a million more questions. And so that's why I still have a job, basically. But I mean, it's why it's really fun to work on these things, because then we have to try to figure out the next thing. That's a great question. Now, an even better question that you should be asking yourself is, what the heck does this have to do with Winnie the Pooh? I promised you a long, long time ago that we would be talking about Winnie the Pooh. And you've been patient with me, and I appreciate that. Yes, this was what I promised. One of the other main lines of research in our lab is we study the morphological effects of captivity. This is not super popular. I love captive research facilities. I love the Lemur Center. We've established that. I love the North Carolina Zoo. The fact is that colleagues of mine, people like me who study the anatomy of animals, like if we go to museums, for instance, they tend to exclude zoo animals from their samples. And they do this because we have kind of known that zoo animals are screwed up. Not really like fun to talk about, but no one had really quantified it. Very few people had quantified, what do we mean by the fact that they're screwed up? In what way are they screwed up? How screwed up are they? And why are they screwed up, right? So if you ever go to the North Carolina Zoo and you look at the lions there, do they have tigers? No, they don't have Asia. But they have tigers? Do they have Asia?
I know they have tigers there.
Tigers? So if you go and look at their animals, if you compare those animals to animals in the wild, it's going to be very different. But how different is it? Yeah.
Are you talking about animals in captivity who have like a long breeding history of being in
That's
captivity?
a great question. So if we
Then-
wild catch them, or if
Yeah.
you, versus if they're
Zoo animals now are like a long lineage of having been.
essentially all zoo animals. There is very little capture of wild animals being brought into zoos anymore. So practically like well over 90% of any mammal that you've seen in a zoo has been a captive bred animal. But we wanted to know, like, so what is the effect of captivity? In other words, how are the skulls and teeth of zoo animals different than wild ones? And my students and I, we measured a whole bunch of skulls. And we started this project with lions and tigers. And we started them in the collections of these museums. Do y'all know what museums these are?
Smithsonian.
That's the Smithsonian. You can't go there now. They won't see you. They won't even answer my emails, even though my students want to go there. And what's this one?
No, that's the Museum of Natural History in New York.
Yeah, that's the American Museum of Natural History. Great museum. Two of the best museums in the entire world. Fantastic museums, even though they're closed. Don't bother them. And what's cool about these museums, there's a lot of cool stuff about these museums, but one of the things that's cool about these museums is that they have really long relationships with zoos going back over 100 years. So from the very beginning of the Smithsonian and the National Zoo was archiving their dearly departed with the Smithsonian. In fact, they are the same institution, right? The National Zoo is the Smithsonian. You can't go there. They're closed. They'll turn you away. The zookeepers are working. They are proud to tell you that the animals are being fed. Don't worry about that. But you still cannot come. But in the AMNH, they have had a very long relationship with a fantastic institution, the Bronx Zoo. In fact, what is WCS? Is that the Wildlife Conservation
Society, yeah.
Society? So the Bronx Zoo is one of like 3 or 4 or 5 zoos in the New York area, all related to one organization. So the Bronx Zoo, the Central Park Zoo, Prospect Park. So all of these animals for over 100 years, I think, have been going to the American Museum of Natural History. And so we have lions and tiger skulls that's from these captive collections from all the way back in the day. And so what we would do is we would take one of these skulls and we would use what's called a microscribe or a digitizer. And usually this is a device that's used for engineering. It's basically a little stylus on an articulated arm, and you can poke at your thing at specific places and capture the 3-dimensional coordinates of that object. And so we would poke at our skulls in 43 different spots to try to capture the shape of each of these skulls. And we did this for every lion and tiger that we could come to, right? And so obviously we're not getting like this little blue car. Instead, we're getting a digital version of our skull. Unfortunately, when you poke a skull like that, it doesn't look like a skull like this. It ends up looking like a skull like this. So not like super skullish. Maybe you can see it. If you squint, we already have some head turning. So maybe if you squint and turn your head, you can kind of see it like these diamonds there. That's like the eyes, right? And then that's the back. That's the front. These triangles here, those are the jaws, right? And there's like overlapping of those triangles and overlapping of those diamonds because we've captured the left and right eye and the left and right side of the jaw, right? And so this animal is facing off to the right of the screen. And we did this for all of the lions and tigers. And you can see that on different days, we would put our microscribe down and like the base would be in a different position. And so our piles of lions and tigers ended up looking like they were in a different place in digital space. So that's why we get this like crazy thing. So you can see some of them are tightly aligned. Some of them we got like one and then we moved to the device or something like that. And so the first thing that we do is we'll line them up, okay? And we'll line them up in terms of their scale. So sometimes we'll make the small ones, we'll basically scale them to the average size of the sample if what you want to do is establish differences in shape. So you'll align them in terms of scale. You'll translate them. So we'll take all of those clouds and just like line them up from like the midpoint of that whole space. And then we'll rotate them, right? So like if we had like one facing the other direction, then we'll move those coordinates to line up those coordinates. And this is something, so this is done like in this two-dimensional case on like this insect wing. So if you have like the blue one and the red one, the first thing you'll do is you'll make the blue one the same size, then you'll shift it over because it was kind of to the left, and then you might rotate it to line it up. And now we can see that the red one is fatter and the blue one is longer and skinnier, but we are now comparing kind of like to like. And this is something that's called Procrustes superimposition, which sounds really fancy. And like so much in science, or it's named after an old like Greek thing. And so the quick aside is that the Procrustes story is, have any of y'all heard of Procrustes before? Have you heard of Procrustes superimposition? This is a very common thing in biology. I think it's common in like mathematical sets too, like when you're trying to do stuff like this. But Procrustes in Greek mythology was referred to, every time you see something about it, they call him a rogue blacksmith. And he was also known as the stretcher, which is a great nickname. And the way the story goes is that he had an iron bed on which he compelled his victims to lie. So he was literally a blacksmith, but he had a bed. And if the victim was shorter than the bed, then he stretched them to fit the bed. And if the victim was longer than the bed, then he cut off that person's legs or his feet. And so here's like this vessel here where you can see Procrustes with his blacksmith hammer. You can see his metal bed, and he's making this poor guy fit on his bed. And so this is not a very good bedtime story to tell people. So we'll get back to you, Winnie. I apologize. Okay, so anyway. I love Procrustes. Okay, so we found some really interesting stuff. What makes the skull that shape? So we were trying to figure out whether it was a lion or a tiger, whether it was male or female. We were trying to figure out whether it lived in a zoo or the wild, right? Those are our basic conditions. It turns out that we can easily tell lions and tigers apart, which is great news because my friends would have thought I was an idiot if we couldn't show this, right? Lions and tigers are different animals. Even without the stripes, we should be able to tell them apart, right? And we could. That was the number one thing. So lions and tigers fell into different, what we call morphospace, so different space in our analyses. Very, very big relief. But we also found a surprise. The surprise was that whether an animal was from a zoo had twice as big an effect on the shape of the skull as whether it was a male or a female. This is amazing because lions and tigers are what we call sexually dimorphic animals. A lion and a lioness have different shaped skulls. A tiger and a tigress have different shaped skulls. And yet that difference was half as much as the difference imparted by whether the animal was from a zoo or from the wild. So zoo lions are more different from wild lions than male lions are from female lions. And the same is true for tigers.
How many generations would it take from a wild animal to get into this?
It's a really good question, and it relates to what you were asking about, because the next question is why? What is going on here? And so what you're asking about is that whether we have some kind of genetic thing going on. Maybe we've released something in evolution, or maybe, deleterious things are happening. The thing is that the older animals were not necessarily more wild shaped than the more new animals. Like, so it basically correlating the difference between wild and captive animals to their time, the length that taxon has been in captivity, that didn't have an effect on the shape of the skull. Yeah.
It's just lifestyle and diet. If you looked at me compared to Homo sapien 10,000 years ago, are you gonna be like, wow, that's a modern human, he sits at a freaking computer all day?
Yeah, like that's what, like our pathetic legs and stuff, right? And our pathetic mouths are because we eat McDonald's food too much, right? Like you know, we just ate. Yeah, so the answer is yes. So in other words, zoos seem to mess up the shape of the skull, and we're getting at the question why. And that is the answer. This is what we think is happening. That we think that it's really, so yes, we see postcranial things. So we see like the skeleton changing in captive animals. We think that that has to do with like they're walking on different types of substrates and maybe they're not walking as much, or maybe they're walking repetitively, etc. But when it comes to the shape of the skull, what we noticed is that the points that were most affected by captivity, so this diagram here is called a lollipop diagram, and the little sticks and candies is what the dots and the sticks are called. The ones that are longest are the ones that are most affected by captivity. And it turns out that the ones that are longest are the places that the muscles of mastication attach to the skull, right? And so the parts of the skull that are changing based on captivity are related to the way that the animals are processing the food. And that's because when we first did this research, it was almost universally true that the best zoos in the world, the best zoos, especially in the United States, they insisted on feeding their animals predominantly what is called the Toronto diet. So the Toronto Zoo literally makes a diet that's a complete diet for big cats. And it is literally chubs. They call them chubs. It's like a big tube. Like if you go to the supermarket and buy like sausage meat, you know, that like it comes in like a tube, kind of like that cookie dough tube, right? But this is like full of meat. That meat is horse meat in the Toronto diet, and it's ground up and it's supplemented with vitamins. I'm always asking people about this diet, like what part of the horse is different than like the zebra that the lion is eating in the wilds that you need to add more vitamins, but that's a whole other conversation. But the bottom line is that they grind up this horse meat so that it meets all of the nutritional components that is necessary to keep that animal alive. But it has very different mechanical properties than the animals are eating in the wild. And one of the really vindicating things about presenting this work, and one of the scariest talks I ever gave was at the AZA conference, at the Zoo Association conference. And people were really mad about this, but I was imploring them, please think about carcass feeding your animals. They're scared because they don't want people to see the zoo animals eating another animal. In fact, we don't like to think about eating another animal. Like, you know, when you're eating that sausage that you just ate, you don't think about the cute pig that it came from. I'm sorry I pointed to you and then talked about what we were eating. But Americans have this like real aversion to thinking about, you know, the animals that we eat or the animals that your beloved other animals are eating. And so they're scared about that. And the most horrifying comment I got was somebody came up to me and said that we don't want our animals to be wild, like wild animals. We want them to be docile and like, basically they don't care that they're not morphologically similar. And I had to sit in like a dark room for a while and just think about like reevaluate my entire life after that. But since I gave these talks, thankfully, they've changed and they've started carcass supplementing animals more often now, and in great places like if any of you have ever been to Carolina Tiger Rescue, so out in Pittsburgh, they almost exclusively carcass feed animals. So they'll leave like a whole quarter of a deer out there and the cat will carry it around and chew on it, and they'll tear off the skin and sometimes do gross stuff like sleep with like a rotting skin for like a week and then before somebody takes it away. But like they're doing like this naturalistic type behavior with their jaws, chewing on things. And so thankfully our best zoos are now taking this more into account. And so the first paper that we published on this was on that lion and tiger skull shape. We then started studying, I always forget that I have to qualify. For me, BFFs are black-footed ferrets. My students think of their best friends forever, but my best friend forever is a black-footed ferret. And these are really interesting animals because we were trying to get at that inbreeding effect that you guys were asking about, right? Because the black-footed ferret, this is a species that almost went extinct. And in the 1970s, they tried to round up 100% of the remaining individuals. And unfortunately, that was 13 that they could snatch up. and only 7 of them bred. And so they brought them into captivity and bred them back. And in fact, some years ago, they started re-releasing them into the wild. It's not a super successful story, but now there's a stable captive population at least. But they were crazy inbred, and so we wanted to know, does that inbreeding effect seem to play out in the skulls? And it turns out, so first we studied like their oral health, and so we looked at like the calculus, like the crud growing up on the teeth. We looked at gum disease and tooth loss and stuff like that. And what's crazy, you would think that captive animals have like nicer teeth because like they have literally dentists come and like clean the teeth of a tiger or something. But captive animals have the worst teeth. They're horrible. And it's because they're eating that really soft diet. It's like if you have a dog at home and you give it like those greenies or like chew things, you know, to try to clean their teeth. Like that's you don't need to do that if you fed it like a deer. I'm not saying feed your dog a deer, but you get the idea. They're not getting that kind of cleaning of their teeth. And so captive animals have devastating stuff. And what's amazing, so the next paper we looked at the oral health in the lions and tigers, and we found that the ones that have horrible oral health also have the most screwed up skull shape and vice versa. We found that with the black-footed ferrets as well. This is one of these ferrets, and you can see like the jaws, like the teeth are literally falling out of the skull. These teeth are already gone, and this skull is really warped in its shape. And so that was the last study that we did, was the skull shape of those captive ones. We're doing more now. We're looking at actually herbivores and seeing if captive herbivores are also affected. But what was amazing, I'm trying not to show too many like data bivariates, but I feel like I've showed a lot, so I apologize. But this is a really neat one. So this blue circle here, these are the wild black-footed ferrets. So we have like at museums, we have when these things, before they were nearly extinct, the Smithsonian like shot a few and put them in the museum. So there were a few. I don't think that contributed to their near extinction. And so the wild ones occupied a very tight morphological area. So we call this morphospace, like the size of these circles. That means that they're very homogeneous. And then the ones that were early captive, so the ones from like 100 years ago at the Smithsonian, they occupied a similar space, but they were broader in morphology. So they were kind of like, some were wider, some were longer, some were, but they were in that realm. And on average, their averages, those like plus signs, like kind of almost like line up, right? But what's amazing is that when they brought them into captivity and started trying to breed them back into existence, they started occupying a really, really different morphospace, right? So they fell out in this direction, and we broke it down into two different recovery phases, back when they were trying to breed them back into captivity, and then back when they were trying to get them ready for re-release. And it was kind of disastrous. And what's truly remarkable about this is not just that the skull shape changed so much, but it changed entirely related to like other species of ferrets. So this animal here, this is called a steppe polecat. This is the European polecat, which is like the wild version of like a domestic ferret. But what's amazing is that these captive black-footed ferrets were more different from wild black-footed ferrets than wild black-footed ferrets were from this steppe polecat, right? And so skull shape, that's like if we found that the tiger skulls looked more like a jaguar than like a wild tiger. That's basically what ended up happening here, which was pretty disturbing. So, yes.
Oh, I was just gonna ask, does the morphology typically change over the life of that ferret or after generations of?
A great question. Yeah. So we haven't done, that's what's called like an ontogenetic study. So when you look at the juveniles and then getting into the adults, and we do this for a lot of our anatomy, we haven't done it with the skulls because thankfully, like there aren't enough babies that are like dying. So actually you get a lot of like babies, like stillborns and stuff, but you don't get like a lot of teenagers die and, the equivalent of teenagers die in the zoos. But what we do know is that the young ones look more similar to the wild shape, and it is over the lifetime of the animal. And it doesn't seem, we don't think that it's related to that genetic drift that we're seeing. My recent PhD student, for one of her dissertation chapters, she looked at brown bears, black bears, and sea lions. And the thing that was cool about that is that sea lions eat fish in zoos and fish in the wild. Right? And they don't really chew at all. They just kind of like snatch it up and swallow it. And so the idea was that if diet was driving this, then the bear, and bears have historically been fed literal trash, like bread and like whatever. Like a bear will eat anything. You know a bear will eat anything because they'll eat your trash out of your backyard, right? And so the idea was that the bears would be more screwed up than the sea lions. In fact, she found bears were very, very screwed up, more screwed up than the lions and tigers, but the sea lions were also really strange, and we haven't figured it out. And it doesn't pertain to their masticatory muscle attachments. So there's something else that was released. But yeah, it doesn't seem to be genetic. It seems to be something that happens over the life of the animal, and we're still working. Because I give this talk for like kids, I try to give like the takeaway message. So what does this have to do with Winnie the Pooh? Oh no, I'll get back to Winnie the Pooh, then I'll give you. So Winnie the Pooh, what does this have to do with Winnie the Pooh? Have any of you ever heard of this book, Finding Winnie? It's a great book, Caldecott winning book. That means it's a great book. It's a beautiful book. It's an illustration, and it's by the, I think it's like the great-great-granddaughter of A.A. Milne, the author of the Winnie the Pooh series. And what this book will tell you is that Winnie the Pooh was a real bear. Did you know Winnie the Pooh was a real bear? Anybody? Nobody. You knew Winnie the Pooh was a real bear? This is Winnie here. And this is Winnie with her owner, Lieutenant Harry Colburn. And he was a cavalry veterinarian. This was back during World War I. And so you don't think about it. So the cavalry is like the people who fought on horses. But when you take horses to war, you have to take veterinarians to war. I never thought about that, right? But he was part of the veterinary corps that was taking care of all of these horses. And he was from Winnipeg, Manitoba, so up in Canada. And on his way, they had to stage in Toronto before they flew over, or took a boat. I think they flew over to England. No, they must have taken a boat in World War I. So before they were going over to England. And when he went from Winnipeg to Toronto, in Toronto on the street, somebody was selling 2 black bear cubs. This is not a Canadian thing anymore. Like you don't go to Toronto to get your $20 bear cub. But he got his $20 bear cub in Toronto on his way over. And that bear basically became the mascot of the Cavalry Corps. So this is little Winnie with the other cavalry veterinarians here. And everybody loved little Winnie. Winnie was adorable. But bringing a bear to war is a bad idea. I tell this, but apparently there's like a famous fighting bear from Poland. Have any of y'all heard of it? My students will tell you about the fighting bear in Poland. But in my opinion, and I think it's an informed opinion, bringing a bear cub to war is a bad idea. And so, Colburn did a smart thing. I mean, it was not smart to buy a $20 bear in Toronto. But before he went to war, before he went to Germany, he brought Winnie to the London Zoo. The London Zoo is what we regard as the first modern zoo. Back then, not what you would think of as a modern place, right? This was the era of like iron and concrete and like kind of inhospitable. But zoos were very different back then, okay? Here, this is very cool. So this is from around the same time, actually earlier. Does anybody know what that animal is? No, it's not an okapi. That's a good guess. It's similar color. What's that? Do you know what it is? No, a zorse is a really good guess too, because it does look like half zebra, half horse. Does anybody else have a guess? This is one of those words that you're going to think I made it up if you haven't heard it. This is what's called a quagga.
Oh, it's a quad.
A quagga. It's spelled quagga, GG. So this is an extinct horse. So this is one of these like famous extinction cases that over the last like 150 years. So like the passenger pigeon and the dodo bird. The dodo bird was earlier. The quagga went extinct, I think in like 1930. And the last one died in the London Zoo. So this is a dude, very dapper guy with a top hat looking at his quagga. Not his quagga, the London Zoo quagga. Here is a keeper shaking hands with a sea lion. I don't know what this guy's doing. He's catching his impala mid-air or something like that. These guys, I like to think that they're playing some kind of rock and roll. No, that was before rock. I don't know what they're playing for these wolves, but they're looking so serious while they're doing it. This guy is sweeping his elephant. Yeah, different times, right? This guy's watering his penguin. This is great. You never see people water their penguins adequately anymore. My favorite one is this guy is turtle waxing his tortoises. And I want to show you, I want to point out, first of all, where he's storing his turtle wax while he's waxing his tortoises. And also, They look immaculate. Don't they look great? So again, we don't turtle wax our tortoises. But what was really interesting is at the time, people interacted with the animals much more at the zoo. So even when I was a kid, you could go to the zoo and ride on elephants, although we did not look as fancy as these people when they were riding on their elephants. These ladies are drawing something in the mouth of this elephant. This kid is feeding a giraffe. You can still go to zoos and feed giraffes. You can't go in the place with the giraffes. That's a bad idea, but you can still do this. This is a bad idea. Like I am not in favor. So when I first gave this talk, my daughter was exactly this age, and I was like, do not hang out with an African crested porcupine, not to be recommended. But she looks fine. She looks happy. I have no reason to believe that she died like right after this picture. This is a picture of Winnie the Pooh interacting with a visitor. And this visitor here is a young boy named Christopher Robin Milne, feeding Winnie honey. And yeah, I mean, the outfit is a little like, again, our daughter would say, let him live, right? So, but yeah, this is little Christopher Robin Milne feeding Winnie. And Winnie had a sweet tooth. Winnie loved sweets. And Christopher Robin fell in love with Winnie. And Christopher Robin's father was the author A.A. Milne, the author of the Winnie the Pooh books. And so Christopher Robin fell in love with Winnie and went to visit Winnie regularly at the zoo and brought Winnie treats, sweets every time, and loved Winnie so much that he went home and he renamed the stuffed bear on his bed Winnie. I don't know where the Pooh came from. I don't think the book explains that, but he renamed his stuffed bear Winnie the Pooh after this bear, Winnipeg, right? So the name stuck. Colburn renamed his bear, or named his bear Winnipeg for his hometown. And it was, everybody referred to Winnie as, or Winnipeg as Winnie. What other stuffed animals do you think Christopher Robin had on his bed? So he had a tiger, a little pig, a kangaroo, right? So basically his father ended up writing the stories. They were bedtime stories, like the stretcher who chopped off people's legs. No, that's not a bedtime story, but so he wrote the bedtime stories based on Winnie. And again, what on earth does this have to do with what I was teaching you guys about? Well, this is my favorite headline of all time. This is from the Huffington Post. Winnie the Pooh's skull is here to destroy your childhood. What's better than the headline is the subheadline, oh bother. Oh bother. Because Eeyore is my favorite animal of all of them. And the reason that HuffPost decided to go with this headline is because this is Winnie's skull. Winnie's skull is bad, okay? You don't have to be a biologist to know something terrible is happening here. What is wrong with this skull? Like there's no teeth, right? So Winnie's teeth are gone. Some of them are full on lost. Some are probably ground to the bottom or rotted out. All of this porosity that you see here, that's all gum disease. It's really sad. And of course, the skull shape, you all have to take my word for it, but the shape of the skull is very different than that of a wild black bear. And this is because Winnie was fed a bad diet, right? So people would feed Winnie sweets, and bears love sweets. Bears do eat honey, but they also eat other stuff that is not going to rot their teeth out of their head. And so this is where I give that take-home message for your kids. If you ate nothing but hamburgers and sweets all of your life and nothing to really chew on, your head would be messed up too. And so when your mom tells you to eat your vegetables, we should do that. Thank you very much. Oh, I forgot that information. There we go. Thank you very much. And then here's for Preston. I don't know how long I spoke. Oh, that was like pretty close to an. So I don't know if we have time. We have some time?
If people need to leave, that's fine, but.
Yes.
Question.
And I can stick around a bit as well. Does anybody have questions, comments, concerns? I try not to show too many growth pictures. Yes.
Have you ever had to give a gorilla a Heimlich maneuver?
No, I have had to drive a gorilla 500 miles in a luxury SUV. It was dead. But yeah, no, I haven't done a Heimlich maneuver. They have really deep chests, so that would be very, very difficult to do. And their guts, like when you do the Heimlich, you're trying to like squeeze their guts up. They have giant bellies too, so not to be recommended. Yeah, but my students did ask me. It was like a year ago when everybody was talking about like a man versus 100 gorilla, or 100 men versus a gorilla who would win. Like, but we can talk about that as well. Yeah, that's an interesting question. Have you? I should have.
No, I just was, but that was exactly what I thought when you're talking about giving them big chunks of food to eat. I'm like, if they choke on it, what are you gonna do with a gorilla?
That's an interesting question. So animals, they don't choke that much, right? And actually, it's a weird part of our anatomy that we choke. It's because of our bipedality and stuff like that. But what's amazing is, so another part of my research, this is gonna sound really off the wall, but like my lab, we also study how animals react to solar eclipses. That's a very different thing. And the number one question I got asked, so I was interviewed by 40 different journalists before the eclipse that was in April of 2024. And the number one question I was asked almost every time, does anybody have a guess what I was always asked about? An animal behavior expert about solar eclipses. What do you think people were concerned about? Whether they had to worry about their dog looking at the sun. Right? Only people are dumb enough to look at the sun. So yeah, it is not a concern. The answer is it's not a concern, but yeah, no, that's a great question. Yes.
So you were talking about the fact that maybe some of the AZA people were a little resistant
Yeah.
to feeding carcasses.
Yeah.
But I'm not sure when you gave that talk, but was that in conjunction at all with sort of an increase in understanding about captive enrichment?
So captive enrichment, that's a really good question. So enrichment is what we give animals to increase the variety of their behaviors and with the idea that we are reducing stress in the animal. So when you see what we call stereotypic behaviors, like animals pacing, et cetera, that is interpreted to mean that the animal is not in a good place psychologically. This was very much thought about in terms of the pacing, like the locomotive behavior, so different substrate use, et cetera. A little bit, there were dietary enrichments as well. So like at that time, cats were given like cow bones to chew on, which that's really dumb. Like cows don't chew, or cats don't chew on bones. They eat the meat off of bones. But I don't know why they decided that that was like something that was going to be good for them. And so they were thinking a little bit in terms of enrichment, but what they weren't thinking about was the mechanical properties of the diet, right? In fact, the people who create diets at zoos are called nutritionists. So the position, like if you're hired to make a diet at a zoo, you are going for a job as a nutritionist. And nutrition is really thinking about the chemical components of the diet and not thinking about those mechanical properties. And so thankfully, like I think that we were able to push them. And so now more and more, they're incorporating more. It's very easy. So they literally will mail the Toronto diet to you and you just flop it out into the thing. Like when you open a can for your cat, you just, right? And that's much easier than like sourcing like portions of a deer or, parts of a cow, et cetera. And also you don't have to worry about the visitors being horrified that this carnivore is eating another animal. I don't know why they're worried about that. To me, that seems like an education opportunity, but again, a disturbing extent, zoos are more about entertainment than education sometimes. And so you have to walk that line. Yes.
There's a quick question. You mentioned briefly that you are that your lab was also expanding research of the difference between skull morphology in captive versus non-captive animals for herbivores. I was curious which animals or which herbivorous animals that.
That's a great question. So my students are studying giraffes because we have good like wild and captive ones. They're looking at pronghorn antelope. Again, we have good wilds. I think they were looking at tapirs. And so they were trying to get ones that are very distantly related to each other to try to see if their skulls are affected in different ways. With the idea that like, even though they're all going to be eating forms of grass like hay, alfalfa, et cetera, maybe the captive one is drier than like wild graze or something, and maybe that has some kind of effect. So we're hoping to collect the second part of the data in January, but stay in touch, stay in tune.
All right, well, I think we'll close it so people can view, but
Leave without feeling rude.
if you want to come up and ask questions, you can.
Yes, thank you so much. And that's my email address. Feel free to contact me.